Welcome. Today we're going to be talking about cell division and reproduction. This section has four major ideas. The first one is why cell division and reproduction needs to happen in the first place. After that, we'll talk about specifically how eukaryotic cells go through two different types of division, mitosis and meiosis, and why each of these are important. And finally, we'll talk about what happens when either mitosis or meiosis goes wrong. First, we'll start with basic cell division. When we talk about cell division, we're thinking about how cells reproduce one another. This is the heart of all reproduction across all organisms and is the basis of cell theory, the idea that cells originate only from pre-existing cells. Now, when we think about cell division, we often think about reproduction only, but that isn't the only thing that we use cell division for. For example, we also use cell division to heal. Cells have to reproduce themselves when they're part of a multicellular organism to allow them to replace damaged tissue. So when you get cut or a starfish loses its arm, it needs to use cell division to replace the part that's missing. In addition to that, organisms change over time. You weren't always the person you are today, were you? Of course not. You started out as a single cell, a fertilized egg, and then 2 cells, and then 4 cells, and then 8 cells. Sometimes cell division is important for growth and development, rather than reproduction itself. Now, there are 2 types of cell division. The first one is asexual reproduction. This happens when a cell produces an identical daughter cell. The other option is that a cell can reproduce sexually. This is important for producing cells that are different from the original. For example, if we were to think about a family like the one in this picture, we'd notice that these people are pretty different from one another. They have different skin colors, they have different hair, they have different eye colors, they're different heights. This variation comes from sexual reproduction and relies on the idea that the cells produced this way are unique. We'll spend some time talking about sexual reproduction later when we get to meiosis, but for now, let's focus on asexual reproduction. Asexual reproduction happens in a variety of ways, but right now we're going to focus on the simplest ways that this can happen. The first is very popular for bacteria. This process is called binary fission. Essentially, what's going to happen is that the cell will grow until it gets large. We talked about how cells have a limit to how large they can be. At that point, the cell needs to divide before it will cross that limit. The cell will replicate its DNA, which in this case is in a nucleoid, because it's bacteria. Then, the cell wall in the plasma membrane will constrict in the middle of the cell. This is an EM picture of that happening. You can see that there is an indent in the dark red areas, which are part of the cell wall, as well as in the plasma membrane. Eventually, this constriction will reach all the way through the cell, separating the 2 copies of DNA. When this happens, the cells will then separate. And we're done. 2 identical cells. If we were to look at the entire bacteria while it's going through this process, we can see that the nucleoid was divided into 2 parts. That's the gray area here. Bacteria aren't particularly picky about how much of their DNA gets in either cell. As long as there's at least 1 copy of their circular chromosome, that's enough. The other option for prokaryotic cells, and some rare eukaryotes, is budding. Budding is pretty similar at the beginning. The nucleoid is going to replicate, so the DNA gets copied. But instead of dividing down the center and creating 2 equally sized cells, the cell pushes out a piece of its membrane to create a bud, and pushes the nucleoid into that bud. Eventually, when the bud is large enough, the cell will then constrict and it will become 2 separate cells, the larger parent cell and the smaller identical daughter cell. Now, I mentioned that eukaryotic cells sometimes do this too. Particularly, that's common for yeast. It works the same way, except for the yeast cell is going to replicate its nucleus near where the bud is formed. Same idea though. Now, the process of cell division that we've talked about so far is the very basic, most easy level of thinking about it. The cell gets big, replicates its DNA, and cuts itself in half in one way or another. Now we're going to focus on talking about how eukaryotic cells go through this process. Eukaryotic cells have specific numbers of chromosomes that they rely on, and they need to make sure that all the chromosomes are exactly where they belong. This means it's a little more complicated than just cutting yourself in half. Before we can really talk about how chromosomes behave, we need to talk a little bit about their structure. So we know that chromosomes are made up of DNA, and that DNA is a double helix made of nucleotides. What you might not have known is that DNA is actually more than that. We can kind of think about DNA like going to the yarn store. If you have a ball of yarn, it's a lot easier to carry that yarn if it's all wound up into a ball. It's a lot harder to carry it if it's all unwound. Your DNA isn't much different. Your DNA is going to be wrapped around proteins called histones, and those histones will be wrapped into coils. Those coils will further coil into supercoils. And these supercoiled materials are what we call chromatin. So DNA itself is just the nucleotide double helix, but chromatin is packed DNA wrapped around histones. We can actually see this when we look at condensed chromosomes, things that are tightly wrapped. These are Drosophila chromosomes. Notice that they are linear. Prokaryotic chromosomes are circular. The banding pattern of these will also be important later. If we were to look at them in the context of a nucleus, this is what they would look like. Now, do you know how many chromosomes a human being has? It's okay if you don't. The answer is 46. Human beings have 23 pairs of chromosomes. You can see this in the karyotype right here. A karyotype is just where chromosomes have been laid out next to each other. This particular karyotype belongs to a male. We can tell by looking at the 23rd chromosome, where there's one X chromosome and one Y chromosome. Now, all of these chromosomes come in pairs. There's a special name for that. These are homologous pairs. Homologous pairs have the same information on them. They might encode the color of your eyes, for example, but they're not identical. One might say that your eyes should be green. The other might say that your eyes should be brown. The reason you have two is because one of each will come from one of your parents. So in this example, the blue are coming from your father. They're the paternal copy. And the purple are coming from your mother, the maternal copy. Before the cell can divide, it first has to replicate its DNA. When we think about chromosomes, we often think about them in this X configuration. But chromosomes normally are linear. In the center of the chromosome is what we call a centromere. When replication happens, this copy of the chromosome will still be attached to the centromere. These identical copies are called sister chromatids. Sister chromatids are identical, whereas homologous chromosomes are not. So the basic idea here is that we'll start with chromosomes, which are DNA molecules. We'll duplicate each chromosome to form sister chromatids. They're going to look like our actual little X connected by the centromere. And then we're going to separate these identical sister chromatids into each of the two daughter cells. Now, it's not quite as simple as that. There are a lot of phases to mitosis. The largest and perhaps most important phase is interphase. Interphase itself is also made up of multiple phases. G1, the first gap, S phase, where DNA synthesis occurs, and G2, the second gap. The amount of time that a cell spends in mitosis is actually quite small, and this is called the mitotic or M phase. M phase itself is also made up of multiple phases, so let's take a closer look at what's happening during each one. Cells spend the majority of their time in interphase. In interphase, DNA is decondensed and fills the entire nucleus. Individual chromosomes cannot be seen. You can see in the nucleus above that it is entirely blue with no discernible or noticeable chromosomes. At the same time, centrosomes exist but are not yet growing to create a mitotic spindle. The centrosome is a special organelle that only exists in animal cells. When we talk about mitosis and meiosis, we'll be focusing primarily on these types of cells. The centrosome is important because it is made up of microtubules that are going to guide where chromosomes will go during the process of mitosis. During interphase is also when the cell is going to replicate its DNA. This happens during S phase and is critical for mitosis to be successful. After interphase, the first phase of M phase, or mitosis proper, is prophase. During prophase, the centrosomes will grow, forming the mitotic spindle, which is a connection of microtubules. Chromosomes will start to condense and become visible within the nucleus. If you look closely at the cell above, you can see that instead of being uniformly blue, there are little squiggles inside. You can also see that the mitotic spindle is starting to form, those long, thin green projections that are coming from where the centrosomes are located. Now, your textbook divides prophase into two phases, early prophase and prometaphase. During prometaphase, this mitotic spindle will start associating with kinetochores, which are located on the chromosome, and the nuclear envelope, which keeps the chromosomes inside the nucleus, will break down. For our purposes, we are going to combine prophase and prometaphase. So for us, in prophase, chromosomes condense, the mitotic spindle forms and associates with chromosomes, and the nuclear envelope breaks down. The next phase is perhaps the most iconic phase, and that is metaphase. During metaphase, the mitotic spindle applies pressure to the chromosomes, which causes them to line up in the center of the cell. You can think about this like an object with strings tied to it. If you hold that object, you can suspend it evenly between your hands by the strings. That's what's happening here. Metaphase is relatively short. As soon as all the chromosomes are lined up properly, it will end. The following phase is even shorter, and that's anaphase. During anaphase, the centromeres, which hold the two sister chromatids together, are going to break down, allowing the chromosome to separate and the two sister chromatids to start to move toward their sides of the cell. Once they reach their side of the cell, that will be telophase. And when that happens, the cell will begin to divide in what is known as cytokinesis. Now, cytokinesis occurs differently in plant and animal cells. In animal cells, a cleavage furrow will form. That cleavage furrow looks kind of like a ring around the cell. You can think of it like a drawstring bag. As you pull the ring, or the string, it causes the cell to pinch off until the two cells separate. If you're having trouble remembering about the cleavage furrow, you can remember that it looks kind of like an old person's butt. That's what I used to think about it. Now, if we think about how plant cells might work, how are plant cells different from animal cells? Well, when it comes to dividing, the biggest difference is that they have a cell wall. The cell wall provides a problem. If you were to tie a string around a cell wall and pull, you wouldn't be able to pinch the cell apart. Instead, you have to create a divider from the inside. So what's going to happen is that vesicles containing extra wall material are going to line up in the center of the cell. They will then fuse to create what we call the cell plate, and the cell plate will move outwards until it divides the two cells in half. So animal cells divide from the outside inward using a cleavage furrow, and plant cells divide from the inside outward using a cell plate. If you want to watch cells divide, I've included two different videos that you can watch. It's worthwhile to try and see if you can recognize all of the major phases. It's going to be very difficult to tell the difference between telophase and cytokinesis because those things happen roughly at the same time, but it's still good practice. Now we're going to talk a little bit about how we regulate cell division because the regulation of cell division is extremely important. The first thing to keep in mind is that cells do not randomly divide. What normally occurs is that when you grow cells, for example in cell culture, the cells will divide until they reach some signal that tells them to stop. In the case of your skin cells, for example, they will continue to divide until they have created a single layer of cells. If you were to remove some cells, they would then divide to fill that space. The only time that cells will randomly divide and ignore these kinds of signals is when there's something wrong with them. This is normally the case in cancer cells. Cancer cells are either unable to detect the stop message or they have an overactive divide message. Either way, they are behaving abnormally. Generally, there are three major places where cells will check to see whether or not cell division should occur. These are the G1 checkpoint, the G2 checkpoint, and the M checkpoint. If we think about what's going on at each of these time points, we can get a good idea of why these checkpoints exist. For example, the G1 checkpoint takes place during the G gap phase, but before DNA replication has occurred. This is a good time for the cell to check, have I gotten any messages that tell me I should prepare to divide? If I have, I should replicate my DNA. If I haven't, I should stay in G1. Once the cell has replicated its DNA, it needs to decide whether or not it should enter M phase. This checkpoint at G2 determines whether or not the cell should continue with the process. Has it gotten a signal to tell it to actually divide? Some cells replicate their DNA because they need extra chromosomes, but they never actually divide. The other thing the G2 checkpoint is for is to make sure that DNA replication has occurred properly. If it hasn't, the cell can abort going into M phase, take apart the synthesized DNA, and return to G1. The last checkpoint is at the end of M phase, before cytokinesis occurs. This checkpoint primarily exists for the cell to check and make sure that cell division has occurred properly. If it hasn't, it can try again. Generally, the messages that cells will use to determine whether or not they should pass through a checkpoint will come from one of two places. They will either come from outside the plasma membrane, in which case we call this a signal transduction pathway, one where a receptor that comes from outside interacts with a membrane protein, which interacts with other proteins in the cytoplasm. Or they might come from inside. An example of one of these would be growth factor. When your cells are exposed to growth factor, this tells them that you are in the process of growing and that they need to divide. This allows them to bypass the G1 checkpoint and start to replicate their DNA. These kinds of signals rely on a wide number of proteins, which we call relay proteins, to make sure that they occur properly. This is why it's so easy to have problems with growth, and why it's one of the most common types of developmental abnormalities. Now we've completed talking about asexual reproduction, it's time to think about sexual reproduction. In animal cells, sexual reproduction happens through the process of meiosis. Your body has two different types of cells. It has somatic cells, or body cells, and it has sex cells. Sex cells make eggs and sperm. All of the cells in your body contain a specific number of chromosomes. For example, as I mentioned earlier, human beings have 46 chromosomes. Homologous pairs of chromosomes contain the same basic information. This is because all chromosomes carry genes that are located in specific locations on the chromosome's body, called a locus. Most chromosomes are autosomes. These are chromosomes that are not associated with determining your sex. The only exception would be the X and Y in humans. Those would be sex chromosomes. Cells that have two sets of chromosomes are called diploid. Gametes, like eggs and sperm, are haploid cells. That means that they only have a single set of chromosomes. Some species, particularly plants, can have even more copies, as many as 5 or 6. But for our purposes, we will be spending time thinking about diploid cells and haploid cells. So one of the big questions is, how do we go from a diploid to a haploid cell, and why would we need to do that? The answer to that question is meiosis. Meiosis is the process of producing non-identical haploid cells from a diploid parent. This is how we make eggs and sperm. On the surface, the phases of meiosis are very similar to what they are in mitosis, and they have very similar names. One of the things that we're going to be keeping track of in this particular set, though, is how many chromosomes each cell has. We're going to use a shorthand for that. Down at the bottom, you'll see that it says 2n equals 6. This means that this cell is diploid, 2n, and that the total number of chromosomes that it has is 6. This means that it has 3 homologous pairs. The first phase is interphase, just like in mitosis, and the cell looks pretty much the same. The chromosomes are all decondensed, they're inside the nuclear envelope, and the centrosomes have not yet started to grow. In the beginning of prophase 1, the first round of meiosis, instead of just condensing, the chromosomes are going to find their homologous pair. These pairs are then going to hold hands, essentially. They're going to form little crossover events that are going to hold the pair together. This forms what we call a tetrad. A tetrad is a connected pair of homologous chromosomes. The spindle will also start to grow, the same way that it did during prophase of mitosis. During metaphase 1, the chromosomes will line up, but this time, instead of lining up single file, they'll line up as pairs because they are attached to one another as tetrads. Instead of having the centromere separate during anaphase 1, the tetrads will break apart, and each whole chromosome is going to migrate to its side of the cell. Then there will be a brief phase in telophase and cytokinesis where the cell will go ahead and divide. At this point, there is now only one copy of each chromosome in the 2 daughter cells. This means that instead of being 2n equals 6, we are now 1n equals 3. That makes these cells haploid. This is sometimes confusing for students because they look at it and say, well, but there are 2 sister chromatids. Doesn't that mean that there are still 2 copies of every chromosome? It could mean that, except we're missing the other copy. We only have the maternal or the paternal copy, not both. So it's a good idea to count the number of chromosomes based on the number of centromeres that are present. In this case, each of these has only 1 centromere, so it counts as only 1 chromosome. Because we've gone from diploid to haploid, this is called the reduction step. Now we need to make sure that we have the correct number of sister chromatids per cell. So we're going to go through all of this division again. We're going to go through a new prophase, where the spindle will start to regrow and the nuclear envelope, which was briefly present during telophase and cytokinesis, will break down again. This time, when we enter metaphase, the chromosomes will line up single file, the way they did in mitosis. The centromere will break down, allowing the sister chromatids to separate in anaphase 2. And in telophase 2 and cytokinesis, the cells will separate again. Notice here that instead of having 2 diploid daughter cells, we have 4 haploid daughter cells at the end of meiosis. The reason why we have to do this is pretty simple. In order to be a human being, you have to have 46 chromosomes, and you have to have the right combination of them. If your cells were to just fuse together, you would take 46 chromosomes and another 46 chromosomes, and suddenly you'd have 92. I don't know what that would be, but it wouldn't be human. So instead, if we want to maintain 46 chromosomes and we want to be able to have an egg and a sperm come together, we need to make sure that they only give half as many chromosomes. So the process is going to constantly go between you, the adult diploid organism, which has 46 chromosomes, to your sex cells, eggs or sperm, which are going to have half as many and be haploid, so that when they fuse, you get 46 again. All right, so let's make some comparisons between mitosis and meiosis. First,
Ripley,
both mitosis and meiosis
now.
begin with diploid parent cells that have chromosomes that have replicated during interphase. At the end, mitosis is going to produce two genetically identical diploid somatic daughter cells. This means that they will have two copies of every chromosome and that those copies will be identical between the cells, and that these cells are body cells. That's why they're somatic. During meiosis, we're instead going to produce four genetically unique haploid gametes in the forms of egg or sperm. So if we were to start with a parent cell that's 2n equals 4, that means that it has two homologous pairs. The first step is going to be prophase. In prophase, chromosomes are going to condense and the mitotic spindle will start to grow. In mitosis, the chromosomes will not associate with each other, but in meiosis 1, the chromosomes will find their homologous pair and form a tetrad. In metaphase, chromosomes will line up on the metaphase plate. In mitosis, they will line up single file, but in meiosis metaphase 1, they will line up in pairs because they are forming a tetrad. The process of anaphase, telophase, and cytokinesis are pretty much the same. In anaphase, chromosomes will separate, in telophase they will reach their ends, and in cytokinesis the cell will divide. In mitosis, anaphase will happen when the centromere breaks down, allowing sister chromatids to separate. But in anaphase 1 of meiosis, because chromosomes are tetrads, instead the homologous pairs will separate. Sister chromatids will stay together. Mitosis is done after one round of cell division and results in two daughter cells that are identical to one another and are diploid. 2n equals 4. However, meiosis needs to go through a second round of cell division called meiosis 2. Meiosis 2 looks almost exactly like mitosis and goes through the same phases: prophase, metaphase, anaphase, and telophase. Unlike in metaphase 1, in metaphase 2, the chromosomes will line up single file, and this will allow sister chromatids to separate. At the end of this second round of cell division, we will be left with four unique haploid daughter cells. Alright, so now that we've talked about the two types of cell division, asexual cell division in mitosis and sexual cell division in meiosis, how do we get from all of that to this? Where does variation come from, and how does sexual reproduction help that happen? Well, there are a couple of places that this can come from. The first one is something that we call independent assortment. This relies on the idea that every time chromosomes line up in the form of a tetrad, they could line up in any way. For example, we might have a situation where all of the blue chromosomes end up in the same daughter cell, whereas in another situation, some of the blue chromosomes and some of the red chromosomes might be mixed up. The more chromosomes you have, the more possible combinations you can get. There's a formula that we can use for this, which is 2 to the n, where n is the number of homologous pairs. So you can see pretty quickly that when you have as many chromosomes as you do, 23 pairs, that already gets us to more than 8 million possible combinations just for making eggs and sperm for a single person. And of course, we have to add an egg and a sperm together. So actually, the likelihood of any possible outcome is 1 in 64 trillion. Now, I've heard people say you're 1 in a million before, but I guess they were underestimating. So why does this matter? Well, it has to do with the differences between homologous chromosomes. So I told you that these chromosomes carry the same information in that they might decide what color your eyes are supposed to be, but they might have different ideas about what that color is. Maybe one of them says green and the other one says brown. This means that there are different possibilities for what each egg cell or sperm cell might convey to offspring. Let's think about this in the context of mice. Let's say that there is a chromosome that has instructions about two different things. The first thing it has instructions for is what color your coat is going to be, whether or not your coat is brown or white. And another location on the chromosome determines what color your eyes are going to be, whether or not they are black or pink. We talked about how homologous chromosomes make tetrads during meiosis. This means that during meiosis 1, one of the chromosomes will go into one cell and the other homologous chromosome will go into another. Now, in our example here, the brown and black are on the same chromosome, and the white and pink are on the homologous chromosome. This means that when they separate, they will either give the information for black and brown together or the information for white and pink together. Which one you get is just going to be determined by chance. Now, to make things a little more complicated, there's another way that we can increase our variation, and that's called crossing over. Crossing over is an exchange of corresponding segments between non-sister chromatids. This happens because of the way tetrads form. When a tetrad forms, there are chiasma, or crossover sites, that help hold the tetrad together. Remember, we had talked about them kind of holding hands? When the chromosomes are pulled apart during meiosis, this can sometimes break the chiasma, exchanging parts of the chromosomes with each other. So how does that lead to variation? Let's think about our mouse experiment. The red chromosome contains information for dark coat and dark eyes, whereas the blue chromosome contains information for white coat and red eyes. If a crossover event happens between these two genes, then they might be exchanged. This would result in 4 possible different combinations: 2 that look like the original parents, dark with dark eyes, light with pink eyes, and 2 that are recombinant or in between. For example, the second mouse has a dark coat and red eyes, whereas the third mouse has a white coat and black eyes. Something as simple as where chromosomes end up through independent assortment, or whether or not a crossover event happens, like in this mouse example, increases the amount of variation that's possible. If crossing over is still confusing to you, the PowerPoint presentation uploaded contains a video about crossing over. I recommend that you go and watch it. Alright, so now we've talked about mitosis and meiosis and 2 places that we can get variation: independent assortment and crossing over. Now we're going to talk about what happens when a mistake occurs. This usually will result in a change in the chromosome number or chromosome structure in an individual. The most common thing that can happen is that there can be an abnormal chromosome count. This means that there is either too many chromosomes or too few. We call this type of event a non-disjunction. Non-disjunction events usually happen because of one of 2 things. Either the pair of homologous chromosomes fail to separate during meiosis 1, or the sister chromatids fail to separate during meiosis 2. So how are these things different from one another? Let's say that during meiosis 1 there's a non-disjunction event. This means that one of the homologous sets of chromosomes doesn't separate. This would result in one of the cells having 2 copies of that chromosome, whereas the other cell would have none, and would result in all 4 of the daughter cells being abnormal. 2 of them would have an additional chromosome, and 2 of them would be missing a chromosome. Now let's think about what would happen if instead the non-disjunction event happened during meiosis 2. In this case, the homologous pairs separate normally, but 2 sister chromatids fail to separate during meiosis 2. In this case, the daughter cells of the cell that separated properly would be normal. They would each have 1 copy of each chromosome, whereas the 2 daughter cells that came from the cell where the non-disjunction event occurred would be abnormal. One would have 3 chromosomes and the other would have 1. This might seem like something rather rare, but it actually happens a lot of the time. So let's take a look at what some of the results might be. Now, on this slide, we're looking at a normal human karyotype. Remember, a karyotype is just lining up all of the chromosomes that are present in a cell. If we look at this particular cell, we're looking at a female. We can tell because there are 2 X chromosomes and no Y. This is a normal human karyotype. Now let's look at the karyotype here. Do you notice anything that's different about this karyotype? If you look closely, you'll notice that there are 3 copies of chromosome 21. This is called trisomy 21, and you've probably heard of it before because it's the cause of Down syndrome. The prevalence of non-disjunction events increases with maternal age. That means that women who have children later in life are more likely to have children with Down syndrome. However, the likelihood is still pretty low, only 8% for someone in their 50s. Why don't we see that many non-disjunction events? Well, the vast majority of them are lethal. That means that they result in spontaneous abortion or miscarriage. I mentioned at the beginning that eukaryotic cells are very particular about the number of chromosomes and their arrangement. So it's very important that they make sure that they have exactly the right number. So most of the time, if you're missing a chromosome or have one too many, it's going to result in death. In fact, all autosomal monosomies, which means you have only a single copy of a chromosome, are lethal. There isn't a single one that results in live birth. And there are only 3 trisomies, situations where you have 3 copies of a chromosome, that are compatible with life. And those are trisomy 21, Down syndrome, trisomy 18, Edwards syndrome, and trisomy 13, Patau syndrome. Now, when we say they're compatible with life, we mean they're compatible with live birth. The actual life expectancy for Edwards syndrome and Patau syndrome individuals is 1 year or less. And for severe Patau cases, it's a few days to a few weeks. So making sure chromosome numbers are perfect is very important. Non-disjunction events for sex chromosomes, on the other hand, are a little more lenient. This can result in individuals that have extra X or Y chromosomes or might be missing them. Here are some examples. Individuals that have an extra X but are otherwise male have Klinefelter syndrome. Males with extra Y chromosomes are pretty normal. Females with extra X's, as long as the number stays relatively low, are normal as well. And the only viable monosomy in humans, a single X individual, results in Turner syndrome. Let's look at examples of these. This is a karyotype for an individual with Turner syndrome. You can see that they have homologous pairs of chromosomes for all of the chromosomes except for the sex chromosomes. This individual has only 1 X. Individuals with Turner syndrome tend to be very short, they tend to be sterile, meaning that they cannot have children, and they also tend to have webbed features, such as a webbed neck. This is the only survivable monosomy in humans, and it's relatively mild. On the flip side of this are males with an extra X chromosome, which you can see in this example of a karyotype here. These individuals have Klinefelter syndrome. They usually present as male, but may have female characteristics. They're also sterile, usually have reduced facial hair, armpit hair, and sometimes develop breast tissue. Talking about how sex chromosomes work, especially in non-disjunction events, often leads to the question of what makes someone male and not female. If you're XXY, you have 2 X's, that's female, but you also have XY, that's male. So what are you? Well, sex determination in human beings is actually very complicated. One aspect of sex determination is something called the SRY. The SRY gene is located on the Y chromosome, and it's very important for determining whether or not you're male. In fact, the presence of an SRY is sufficient to cause some parts of male development, and nothing else on the Y chromosome is required for this. That means that as long as you have 1 copy of the SRY locus, you will present some male phenotypes. That's why individuals who are XXY tend to develop as male with some female characteristics, rather than female with some male characteristics. Now, we've talked about how non-disjunction events can happen during meiosis. Can they also happen during mitosis? Yes, they can. This happens when sister chromatids do not separate during anaphase, similar to the meiosis non-disjunction that happens in meiosis too. These are actually relatively common and results in something we call mosaicism. This is the idea that some of the cells in your body are not identical to other cells in your body. Let's use the ideas that we've talked about to think about a problem. How can a zygote, a developing fertilized egg that is male, XY, end up being a Turner syndrome female, XO, when it's born? In this situation, we start with a zygote that is XY. Under normal circumstances, this zygote would grow into a normal male. However, during mitosis, there is a non-disjunction event that gives rise to cells that are XYY and cells that are XO. These are going to grow together to make a mosaic blastocyst, the beginning of the embryo. Eventually, this will lead to an individual who is a mosaic as well. If the majority of the cells that make up the reproductive system happen to be XO, then the individual would present as a Turner syndrome female. Non-disjunctions aren't the only way that chromosomes can be impacted. For example, chromosomes can break, and when that happens, that can result in chromosomes being rearranged. There are 4 major categories that can occur: a deletion, a duplication, an inversion, and a translocation. Deletions are pretty much what they sound like. Part of a chromosome is deleted or removed. An example of this is Prader-Willi syndrome. This is a deletion of the NDN gene. It results in poor muscle tone, short stature, uncontrollable eating, and mild intellectual disability. Prader-Willi syndrome has shown up in a number of popular places, including CSI, where there was an individual who suffered from the syndrome who was under investigation. Another rearrangement is duplication. This is also what it sounds like. A portion of a chromosome is copied, creating a duplicate of it on the chromosome. An example of a disorder caused by duplication is Charcot-Marie's tooth disease. This is a duplication of part of chromosome 17. It results in a loss of muscle tone, constant muscle cramping, and loss of motor functions, and it is incredibly painful. You can see the tensed muscles that cannot relax in the foot to the right. Inversions are a little bit different. In this case, we are essentially flipping over part of the chromosome, so we're changing its orientation. Hemophilia A is often caused by an inversion that's located in FV111. This results in an inability to clot blood, leading to heavy bleeding from injuries. And last but not least is reciprocal translocation. These are relatively rare, but they involve swapping parts of chromosomes that are not homologous. And it's one way that you can have a mild version of Down syndrome. So when we put all of this together, I want you to remember that you are 1 in a million. Well, okay, no, really more like 1 in greater than 64 trillion. Unless, of course, you have an identical twin.